Good morning, everyone. I'm Lory Liza Culaban, and today we will discuss about the microbiology. So, what is microbiology? Microbiology is the study of small living organisms that can only seen by our naked eye. So, the first organism that we will tackle is about the eukaryotic cells. So, what is eukaryotic cells? As you will notice in that picture, eukaryotes are organisms whose cells have a nucleus enclosed within membranes. Eukaryotic cells are larger and more complex. All protists, fungi, plants, and animals are examples of eukaryotes. The protists are one-celled eukaryotes. This group includes protozoa, slime molds, and some algae. The fungi can have one cell or many cells. Plants from simple mosses to complex flowering plants, and also the animals are example of eukaryotic cells. So eukaryotic cells may be either single-celled or multicellular. Eukaryotic cells. Where eukaryotic cells found? It is found in higher organisms such as plants, animals, fungi. This includes molds and yeast. They are characterized by having two nucleus surrounded by a nuclear membrane. They also possess mitochondria, responsible for producing the energy needed for cell growth and repair. Are humans eukaryotic? Despite of the fact that we have gobs of prokaryotic cells living inside and on us, humans are still categorically eukaryotic organisms. This means that all human cells, including those found in the brain, the heart, the muscles, and so on, are also eukaryotic cells. Robert Hooke was the first to discover this cell. So why is eukaryotic cells important? The nucleus is particularly important among eukaryotic organelles because it is the location of cells' DNA. Two other critical organelles are mitochondria and chloroplast, which play important roles in energy conversion and are thought to have their evolutionary origins. The organelles of eukaryotic cells are the ribosomes. The function of the ribosomes is to make protein. The Golgi apparatus to make and process the package of protein. Lysosomes contain digestive enzymes to help break food down. Endoplasmic reticulum, called the intracellular highway because it is for the transporting all sorts of items around the cells. So what is the origin of eukaryotic cells? Eukaryotic cells arose through endosymbiotic events that gave rise to the energy-producing organelles within the eukaryotic cells, such as mitochondria and chloroplast. Mitochondria appear to have originated from an alpha proteobacterium, whereas chloroplast originated as cyanobacterium. Eukaryotes are thought to have evolved between about 1.7 billion and 1.9 billion years ago. So, That's all for the eukaryotic cells. So let's move on to the prokaryotic cells. The scientist who discovered the prokaryotic cells is Anton van Leeuwenhoek. So prokaryotic cells are unicellular organisms that lacks of organelles or other internal membrane-bound structures. Therefore, they do not have nucleus, but instead generally have a single chromosomes, a piece of circular double-stranded DNA located in an area of the cell. It is called the nucleoid. Prokaryotic is from the Greek word means before nucleus. The example of the prokaryotic cells are bacteria. The types of bacteria is the E. coli, Streptococcus bacterium. It is responsible for strep throat. Streptomyces, soil bacteria. Archaea are also example of prokaryotic that are able to survive in very harsh environment. So what is unique to prokaryotic cells? Prokaryotic cells lack an organized nucleus and other membrane-bound structures. Prokaryotic DNA is found in a central part of cell called the nucleoid. The cell wall of a prokaryote acts an extra layer of their protection. It helps maintain cell shape and prevents dehydration. Most prokaryotes carry a small amount of genetic material in the form of single molecule or chromosomes of circular DNA. Prokaryotic cell structure. Prokaryotic cells are about 10 times smaller than eukaryotic cells. Structurally, prokaryotes are very simple cells when compared with eukaryotic cells, and yet they are able to perform the necessary processes of life. Reproduction of prokaryotic cells by binary fissions, the simple division of one cell into two cells. After DNA replication and the formation of separating membranes and cell wall, all bacteria are prokaryotes, as are the archaeans. Embedded within the cytoplasm of prokaryotic cells are chromosomes, ribosomes, and other cytoplasmic particles. Unlike eukaryotic cells, the cytoplasm of prokaryotic cells is not filled with internal membranes. The cytoplasm is surrounded by a cell membrane, a cell wall usually, and sometimes a capsule or slime layer. These later three structures make up the bacterial cell envelope, depending on the particular species of bacterium, flagella or peli, or both may be observed outside the cell envelope, and the spore may sometimes be seen within the cell. So the structures of the prokaryotic cells. The first structure is the cell membrane. So enclosing the cytoplasm of a prokaryotic cells is the cell membrane, or plasma, cytoplasmic, or cellular membrane. This membrane is similar in structure and function to the eukaryotic cell membrane. Chemically, the cell membrane consists of proteins and phospholipids, which serve the same functions, providing rigidity, strength, and protection. The main constituent of most bacterial cell walls is a complex macromolecule macromolecular polymer known as peptidoglycan or murin, consisting of many polysaccharide chains linked together by simple peptide or protein chains. Peptidoglycan is only found in bacteria. The thickness of the cell wall and its exact composition vary within the species of bacteria. So, the slime layers and capsule, or glycocalyx. Some bacteria have a thick layer of material known as glycocalyx located outside their cell wall. Glycocalyx is a slimy gelatinous material produced by the cell membrane and secreted outside the cell wall. There are two types of glycocalyx. One type called is a slime layer. It is not highly organized and not is firmly attached to the cell wall. It is easily detaches from the cell wall and drifts away. The other type of glycocalyx called a capsule is highly organized and firmly attached to the cell wall. Capsules usually consist of polysaccharides, which may be combined with lipids and proteins, depending on the bacterial species. Flagella or flagellum are the thread-like protein appendages that enable bacteria to move. Flagellated bacteria are said to be motile, whereas non-flagellated bacteria are usually non-motile. So next is the peli or the fembrae. Peli or fembrae are hair-like structure, most often observed on gram-negative bacteria. They are composed of polymerized protein molecules called pilin. Peli are much thinner than flagella, have a rigid structure, and are not associated with motility. These tiny appendages arise from the cytoplasm and extend through the plasma membrane, cell wall, and capsule, if present. There are two types of peli. One type enables bacteria to adhere or attach to surface. The other type called as sex pilus or six pilus, enable transfer of genetic material from one bacterial cell to another. The peli that enable bacteria to anchor themselves to surfaces with tissues within the human body are usually quite numerous. In some species of bacteria, pilated strains, those possessing peli are able to cause diseases like urethritis and cystitis, whereas non-pilated strains, those not possessing peli of the same organism, are unable to cause this disease. So lastly, the spores or the endospores. A few genera of bacteria are capable of forming thick wall spores as means of the survival when their moisture or nutrient supply is low. Bacterial spores are referred to as endospores, and the process by which they are formed is called sporulation. During sporulation, a copy of the chromosomes and some of the surrounding cytoplasm becomes enclosed in several thick protein coats. Spores are resistant to the heat, cold, drying, and most chemicals. Spores have been shown to survive for many years in the soil or dust, and some are quite resistant to disinfectants and boiling. So the prokaryotic cell's reproduction is quite simple when compared with eukaryotic cell division. Prokaryotic cells reproduction by a process known as binary fission, in which one cell or the parent cell splits in half to become two daughter cells. Before a prokaryotic cell can divide in half, its chromosomes must be duplicated, a process known as DNA replication, so that each daughter cells will possess the same genetic information as the parent cells. The time it takes for binary fission to occur, the time it takes for one prokaryotic cells to become two cells, is called the generation time. So the generation time varies from one bacterial species to another and also depends on the growth condition. So we're done discussing the eukaryotic and prokaryotic cells. So the next we will discuss their differences. The difference between the prokaryotic and eukaryotic cells. So the structural differences between prokaryotic and eukaryotic cells. Eukaryotic cells contain a true nucleus, whereas prokaryotic cells do not. Eukaryotic cells are divided into plant and animal type. Animal cells do not have a cell wall, whereas plant cells have a simple cell wall, usually containing cellulose. Cellulose, a type of polysaccharides, is a rigid polymer of glucose. Polymer and saccharides. Prokaryotic cells have a complex cell walls consisting of protein, lipids, and polysaccharides. Eukaryotic cells contain membranous structure, such as endoplasmic reticulum and Golgi complexes, and many membrane-bound organelles, such as mitochondria and plastids. Prokaryotic cells possess no membranes other than the cell membrane that encloses the cytoplasm. Eukaryotic ribosomes, referred to as Atis ribosomes, are larger and denser than those ribosomes found in prokaryotic. The fact that 70 ribosomes are found in the mitochondria and chloroplast of eukaryotes may indicate that these structures were derived from parasitic prokaryotes during their evolutionary development, and other differences between prokaryotic and eukaryotic cells. So, in the other hand, eukaryotic cells are larger and more complex than the prokaryotic cells. They have differ in cellular structures of prokaryotes and eukaryotes, include the presence of mitochondria and chloroplast, the cell wall, and the structures of chromosomal DNA. But they have similarities. Both, both cells have plasma membrane, the ribosomes, cytoplasm, and the DNA. The plasma membrane or cell membrane is the phospholipid layer that surrounds the cell and protects it from the outside environment. So you may be asked, are viruses prokaryotic or eukaryotic cells? So eukaryotic cells includes such as microorganisms as fungi, protozoa, and simple algae. Viruses are considered neither prokaryotes nor eukaryotes because they lack the characteristic of living things except the ability to replicate, which they accomplish only in living cells. So, that's all for my discussion about the eukaryotic, prokaryotic, and their differences. I hope you understand, and thank you for watching. Bye!